Good morning everyone. This is the very first time that we are going to talk about the implementation of chlorophyll fluorescent transit analysis to identify the diurnal variations in Dracaena as a stress response. Actually, chlorophyll fluorescence is a newly introduced technique to identify the status of photosynthetic apparatus in plants. In this experiment, we selected Dracaena as the plant species and measured several parameters. Very interestingly, we found that there are certain parameters that show these circadian rhythms in Dracaena as well. Generally, we use the term circadian rhythms to introduce observable oscillations that occur within 24-hour period. In this figure, overall performance of photosystem 2 was illustrated. From blue line you can see a clear unimodal circadian variations in each and every treatment under high light conditions, but it is not identifiable in low light. The specialty in here is always the peak was observed during the dusk. This is a very common feature in camp plants because in camp they keep their stomata opening during the night and fix carbon dioxide to produce oxalate. Then it reduces to malic acids and those malic acids are stored in vacuoles to release the Calvin cycle during the daytime where the stomata are closed. As a result of that, always the performance during the night is higher than day in camp plants. It is a good evidence to say Dracaena sanderiana has camp features too, but it cannot be justified by only looking at the performance index. So we measured another major parameters as you can see in the right side table. While most of them show circadian rhythms, there were some rigid parameters which do not change and they are displayed in pink color. We used two different green colors as there were few deviations in mercury chloride treatment just because mercury has many negative effects on photosynthesis. So ultimately we can conclude that Dracaena sanderiana is a facultative chem under high stress levels, and also chlorophyll fluorescent transit analysis can be used to identify the status of photosynthetic apparatus, and it is a high throughput technique to analyze circadian rhythms in plants.